Hello students, I am teacher Svetlana and in our today's lecture we will learn about sexual reproduction in flowering plants, structure of anther and microsporogenesis, which is covered in the chapter Reproductions in Lower and Higher Plants. The sexual reproductions, especially in flowering plants, it involves fusion of two compatible gametes or sex cells. All organisms reach to the maturity in their life before they can reproduce sexually. In plants, the end of the juvenile or the vegetative phase marks the beginning of the reproductive phase, and it can be seen easily in the higher plants at the time of flowering. Now you can have a look at this image of a flower. The flower is a specialized reproductive structure of a plant in which sexual reproduction takes place. Now what are the functions of this flower? The flower here produces haploid gametes and to ensure that the fertilization will take place. Typical flowers consist of 4 different roles, that is the calyx, the corolla, the androecium and the gynoecium. Students, I know we are all familiar with these parts of the flower. Now, sexual reproduction involves 2 major events. The first one being meiosis and the fusion of the gametes to form the diploid zygote. So, due to this, there is production of genetically dissimilar offsprings. Now, why do we have this variations? As we know that variations are useful from the point of view of the survival and the evolution of species over time. Students, sexual reproduction is characterized by the fusion of the male and the female gametes. This process is called as fertilization. Now, the formation of zygote and then it is followed by embryogenesis. Now, these sequential events that occur in sexual reproductions are grouped in 3 distinct stages, which are pre-fertilization, fertilization and post-fertilization. Have a look at this diagrammatic representation of the life cycle of an angiosperm. This is your flower. Then you have the development of the pollen grains. This is the anther. This is the ovary where you will have the development of the ovum and the egg cells. So these stages are grouped into the category of pre-fertilization stages. Then you have the fertilization that takes place when there is a fusion of the male and the female gametes, and after the fertilization you have the egg that is fertilized, which develops into the embryo, and finally you have the development of the seedling. These stages are grouped into the post-fertilization stages. So each of these stages we will individually see in this chapter ahead. Students, the main reproductive role of the flower is called as the androecium, and the individual members of the androecium is called as a stamen. Now each of these stamen consists of a filament, connective and anther. Have a look at this image here. This shows you the structure of an anther. This is the long filament, you have a connective here, and this is your anther. Students, let us have a look at the structure of an anther. An immature stage of anther is represented by a group of parenchymatous tissues surrounded by a single layered epidermis. Anther is generally dioecious, that is having 2 lobes, and tetrasporogenate. Now, what does this mean? Each monoecious anther contains 2 pollen sacs and dioecious anther contains 4 pollen sacs. So, if you have a look at this diagram, this is a diagram of a dioecious anther. You have 2 lobes, and when you cut open the lobes, you can see 4 different pollen sacs, and therefore it is called as tetrasporogenate. The heterogeneity, that is differentiation, arises when some hypodermal cells get transformed into archisporal cells. Now, what are these archisporal cells? Students, the archisporal cells are the ones which divides into an inner sporogenous cell and outer primary peritoneal cells. Sporogenous cells form sporogenous tissues, and each cell of the sporogenous tissue is capable of giving rise to a microspore tetrad. Parental cells undergo division to form the anther wall layers. So this is the location here in this diagram. This is the location where you have your sporogenous cells. Now these cells are making your sporogenous tissues, which are also called as microspore mother cells. Now, the wall of the mature anther consists of 4 layers. These layers that are there, these layers are called as the anther wall. Now, we have 4 layers for this anther wall. The first layer is the epidermis. It is the outermost protective layer made up of tubular or flattened cells. So, this outermost layer is your epidermis. Then next to it is your endothelial cells. It is subepidermal layer made up of radially elongated cells with fibrous thickening. So the next innermost layer is the endothelium. Inner to the endothelium is the middle layer made up of thin walled cells, 1 to 2 layers, which may disintegrate in mature anther. Then the next innermost layer here is the tapetum. It is the innermost nutritive layer of the anther wall. It immediately encloses the sporogenous tissues. So if you have a look at this diagram here, this is the location of your sporogenous tissue. Just enclosing the sporogenous tissue is the tapetum. It is the innermost and it is the most nutritive layer of the anther wall. Students, in your textbook, there is also an image of a dehinged anther. This is the image that is present in your textbook. So once your pollen grains are ready or mature, the anther wall breaks open and it releases the pollen grains out. So if you have to look at, this is the image of your anther where it is attached to the filament. So this is your anther wall. The anther wall just opens up when the pollen grains are all mature and ready, and you have these pollen grains, they come out of the pollen sac, and this is the lining that you can see of your anther, where this is called as the line of dehiscence, where the anther has opened to release the pollen grains. So this process is called as dehiscence of the anther, that is, release of the pollen grains takes place. Students, let us have a look at the process of microsporogenesis. Each microspore mother cell divides meiotically to form tetrad of haploid microspores, which is the pollen grains. Now, before we move to our microsporogenesis, we will try to understand the structure of the microspore, which is basically the structure of our pollen grain. A typical pollen grain is a non-motile haploid unicellular body with single nucleus. So pollen grains by themselves are not motile, so they require a medium for their dispersal. It is surrounded by a 2-layered wall called as sporoderm. Now this is the image of your pollen grain. This is how it looks in its 3-dimensional form, and this is a 2D image of the pollen grain. The outermost layer, which is called as the exine, is thick and it is made up of complex non-biodegradable substance called as sporopollens. So this is the outermost layer and it is called as the exine. They are very thick in nature. It may be smooth or with a sculptured pattern. This is characteristics of the species. So this outer layer is either smooth or it has these spines present on it. Now it is resistant to chemicals. At some places, exine is very thin, showing thin areas known as germ pores. So if you have a closer look at these exines, there are some regions you will spot out which is having a small opening kind of a structure. So these regions are called as germ pores. Even you can have a look at this 2D image here. You can see that this is your wall of the pollen grain, and in between you have a slight space which is a small opening. That opening is called as the germ pores. Now what are these germ pores meant for? These germ pores are present for the growth of the emerging pollen tube during germination of the pollen grains. So when the germination of the pollen grain will take place, this phenomenon you will see when we will study the development of the male gametophyte. So from this region you will have the pollen tube that comes out, in which there is the transportation of the male gamete takes place. Now the inner wall layer, that is intine, it consists of cellulose and pectin. So the inner wall that is there, which is intine, it's nutritive in nature and it also contains cellulose and pectin. So this is the typical structure of a pollen grain. Thank you students for participating in this lecture. We will meet soon again with the next topic. Till then, take care, be safe and keep learning. Thank you.